Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the transduction. And this is the I explained the transformation process in my previous video, so you just check out that one. And it is the second process by which bacteria transfer their DNA from donor to the recipient. Sorry for the noise. Okay, so here the transduction process is that here the bacteriophage function as the vector. Okay, here the bacteriophage that function as the vector, and this function as vector to transfer the DNA from the donor bacteria into the recipient. Clear? So here the involvement of the phage, bacteriophage, is very important. Without bacteriophage, the donor bacteria, from donor bacteria, the DNA cannot be transferred into the recipient. Okay, and this process is seen in the Salmonella typhimurium. Okay, so here two kinds of transduction process we can see here, that is, one is the generalized process and another is the specialized process. So where the generalized process, the transducing phage that produce the lytic growth, and these are aberrant and contain a random fragment of bacterial genome. So what is saying that? Here the transducing phage that produce the lytic growth are aberrant and contain a random fragment of bacterial genome. So lytic growth is very important here, because one second. So what is happening here? I just forgot to write here one word. That's why I cannot continuing this with proper explanation. Okay. So transducing phage produced here during the lytic growth. So I have already explained about the lytic cycle and the lysogenic cycle in my channel. So you just check out that what is the lytic cycle and what is the lysogenic cycle. So this phage produced only when the lytic growth is occurring, and in that situation a random fragment of bacterial genome produced. Instead of phage DNA, each individual phage carries a different set of closely linked genes. So what is saying that phage DNA that carries with the what kind of genes? A set of closely linked, different set of closely linked genes. That means here the phage DNA is absent. Instead of phage DNA, only one set of different kind of genes are present and representing a small segment of bacterial genome. So what is saying that? That is saying here this process is occurring only in presence of lytic growth, and here the phage virus that carry the bacterial DNA, simple word, that is the bacterial genome, is present within the phage. No phage DNA present here. Clear? And each part of the bacterial genome has approximately the same probability of being transferred from donor to recipient bacteria. So first of all, the bacterial DNA that enter within the phage body or phage cell, and now the actual probability of the bacterial genome is that to transfer their DNA within the another bacteria that are called the recipient, okay, through the phage. And when the generalized transducing phage infect a recipient cell, expression of transferred donor gene occurs. So what is saying that? When the phage that infects one recipient bacteria where must be the bacterial DNA is transferred, so in that situation the bacterial DNA that already present within the fudge, that is transferred now within the recipient bacterial cell, and this is the process of transduction. And here you can see that fudge that acts like a medium. Okay, without fudge, this bacteria that are the donor, they are not capable to transfer their bacterial DNA into the recipient bacteria. Clear? This transduction may be two types. One is the abortive, another is the complete. Okay, another is the complete. Abortive means it refers to the transient expression of one or more donor genes without formation of the recombinant progeny. Simple word that recombinant progeny is absent here. Okay, recombinant progeny is not formed here in this abortive process. And complete process, it is the characterized by the production of the recombinant DNA and the stable DNA obviously, and that inherit donor genes and retain the ability to express them. So here the complete means the recombinant DNA that are very stable, that are produced here, and they inherit the donor, or they are capable to inherit the donor DNA into the recipient one. Okay, and obviously try to express their feature. And in this transduction process, DNA remain double-stranded during the transfer and both strands integrated. Simple word is that both the strands of DNA when enter within the recipient cell, both the strand of the DNA that enter within the cell or within the bacterial cell. Okay. But in case of transformation, that only one strand they are capable to enter within the bacterial cell. But here both the strands must be entered within the recipient bacterial cell. Next one is the specialized. It is the mediated by specific temperate fudge. Obviously here fudge also needed, but specific temperate fudge are very important for the specialized transduction process. And few specific donor genes that can be transferred to the recipient. Not all cases, all the donor genes that are transferred by this process, only few specific genes that can be transferred through this process. And when the lysogen donor bacteria enter the lytic cycle, lysogen donor bacteria enter the lytic cycle and release the fudge progeny, then this process occurs. Simple, the lytic cycle is also needed here and lytic cycle is also needed here. So when the lytic cycle is completed, the release of fudge progeny is now seen, and then this transduction process is occurring. And simple process, the two processes are same, but only the difference is that there is this is doing by the temperate fudge and this is doing by the any fudge, okay, any transducing fudge. And this is possible only to transfer the specific gene, but here it is not needed that specific gene only be transferred from the donor to recipient. Next one is that important features of the specialized transduction cases. The only bacterial genes that can be transduced are those very near the site at which the prophage is integrated. So where the prophage integrated, in that area or that site, the closest DNA present, that only can be entered. Clear? The bacterial cell when infected by the fudge, okay, that area, just this site, at that site, only the DNA present within the fudge body that can be entered into the bacterial cell. And it results from the defective excision of the prophage from host chromosome. So it is another important point that it results only when that from the defective excision of the prophage from the host chromosome. So defective excision means the defective gene cannot be entered in this process. The specialized fudge contain both the host and fudge DNA linked in one single DNA molecule. So here host and fudge DNA both are linked to form one or single DNA molecule, and okay, one second. So here these are very rare case where the lack a part of normal fudge genome and contain part of the bacterial chromosome located adjacent to the prophage attachment site. So what is saying that it is very rare case, and in that situation one part lack a part of normal fudge genome and contain part of the bacterial chromosome. So here the normal fudge genome absence is C. Absence of the normal fudge genome is C, but here the bacterial chromosome that present. So, and which bacterial chromosome is C? Just these are located at the site near the attachment. Okay, so here it is example that formation of lambda gal and lambda bio that causes the loss of lambda genes. So these are the two transduction process, and another transduction process, this is called the co-transduction. So just I have to rub it and then I will try to explain about this co-transduction. Okay. Okay, so this is the co-transduction process. What is the co-transduction process? There is a maximum size of DNA segment that can be packaged by bacteriophage. If two genes are close enough along the chromosome, a bacteriophage may package a single piece of chromosome that carries both the genes and transfer that piece to another bacteria. So what is saying that maximum size of the DNA segment that can be packaged by the bacteriophage in the co-transduction process. And here the two genes which are closely associated or closely remained, they are packaged by the phage and then the transfer of that piece into the bacteria. So in that process, bacterial DNA and also one second, that close enough chromosome may package the single piece of the chromosome that carries. Okay, so here the co-transduction process means here the two, one second. Okay, so what is saying that if two genes are closely remained, then they are easily co-transduced. But if the two genes that are far apart along the bacterial chromosome, so in that situation they will never be co-transduced. Okay, because the bacteriophage cannot physically package a DNA fragment that is too large. So if the two DNA that becomes too large, so they cannot be packaged by the bacteriophage. But if the two DNA, they are closely lined and also small in size, so in that situation they are packaged by the bacteriophage. And this co-transduction can also be used to map genes. So this is used for what? To map the genes that present in the bacterial chromosome. That means it helps in mapping process of bacterial chromosome genes. And the relation of the bacterial chromosome genes by which the mapping process is done, in that situation the frequency, the co-transduction frequency is equal to 1 by d by L whole cube. Okay, so that means C is here the frequency, co-transduction frequency, D is the map distance, and L is the size of the DNA fragment. And here the relation is that frequency of the co-transduction, that is inversely proportional to the distance between the two genes. So co-transduction transduction, why it is called? Because here the two genes that are packaged and behave like single gene and transform into the bacterial cell. But that is the recipient bacterial cell. Okay, so this is called the co-transduction process, and here this is the rough sketch of the transduction process. And just like lysis process also involved in here, and here the infection of the bacterial cell by the phage, and then the transfer of the bacterial genome into the recipient bacterial cell is just this is the rough sketch, and by this process the transduction is occurring. So here the phage is very important and it acts like media. Without phage this transduction is not occurring. Okay, so in my next video I will try to explain about the conjugation process.